pathologist, molecular pathologist from Essen, Germany, and from the West German Sarcoma Center. Warm welcome to the next session, which is dedicated to news and updates and will open a stage to young scientists as well. For the sake of time, I suggest to type short questions directly in the chat. Now we will directly move to the first speaker, to the first presentation, which will be given by Stefan Zöllner. Consider PADI is the request, and honestly, I consider PADI as an international diving organization, but we will learn more about this. Mr. Zöllner, go ahead.
Yes, thank you very much. Can you all see my screen?
Yes, we do.
Thank you. Thank you. Well, hello and welcome to a special event from my side too. My name is Stefan Zöllner and I'm a clinician scientist here working within the cooperative Ewing Sarcoma group in Essen. And I'm very grateful to get squeezed into this lineup of exquisite clinician scientists and to present to you some data of ongoing unpublished projects in our laboratory. So our lab here focuses on translational research, and broadly speaking, translational medicine describes the path which takes an idea towards standardized clinical use through different stages of testing. In the end, what we aim to do is to introduce novel drugs into the current chemotherapeutic backbone to ultimately increase survival for our patients. That's the big goal that we are following up here. And to start, we recently performed an epigenetic drug screen using 142 different drugs. And after 2 rounds of treatment at 2 different concentrations and 5 different cell lines, cell viability was assessed. In our screen, 21 drugs decreased the cell viability by over 18%, at least in 4 out of cell lines. And what I would like to present to you is 2 of these candidates which have been used for further testing. The first one is called Bbcl Amide, and the second is called lestarotinib or SAP701. So Bbcl Amide is an enzyme inhibitor that I first worked with in the lab of Jeff Turetsky. As a collaborative side project that we had with Scott Coonrod's lab from Cornell University. And specifically, Bbcl Amide inhibits a family of protein, which are called PADI. So this inhibitor was further tested and it showed efficacy in a broad panel of Ewing sarcoma cell lines. And in these models, Bbcl Amide reduced colony formation growth while inducing apoptosis in a significant manner. So to get a little bit into the biology of what I'm going to talk about is broadly that proteins can change their structure and function through post-translational modification. So PADI stands for a peptidyl arginine deaminase, and these proteins catalyze a reaction that changes proteins in terms of structure and function. So giving the example of histone proteins, for example, if you have PADI activity on these histone proteins converting arginine to citrullinine, then these histone proteins change their structure and change their function. And ultimately, this histone chromatin architecture gets decondensated. And so the hypothesis is that by decondensed chromatin, transcription factors such as EDOS-5-1 have it easier to bind to DNA and then initiate the transcriptional program that leads to cancer proliferation. So dysregulation of PADI expression and dysregulation of citrullination, which is so now the effect of PADI activity, is implicated in various diseases, including COVID-19, actually, and also cancer. And therefore, we have analyzed different patient cohorts regarding PADI expression and found a PADI2 specific gene upregulation in Ewing sarcoma patients, as shown here. And moreover, looking at different patient samples, we did not only find PADI2 protein being upregulated, but also increased citrullination in patient tumors, although the intensity appears to be heterogeneous. So strikingly, those patients showing a higher degree of citrullinated proteins, which are these images here on the right side, had a worse prognosis compared to those patients that had less citrullination in their tumors. So moving on and using a doxycycline-inducible EWS1 knockdown model from Thomas Grünewald's lab in Heidelberg, we have shown that PADI2 gene and protein expression are regulated through EWS1 presence, meaning if you knock down EWS1, you reduce PADI2 gene and protein expression. And if you knock down PADI2 on the other side, you do achieve a highly decrease in proliferative activity, which is similar to the EWS1 knockdown. And both knockdowns led to the reduction in citrullination, as you can see here on the right side, as marked by the fainter red fluorescence in the Western blot images. So moving on and looking to RNA sequencing data from Ewing sarcoma patients, we found that PADI2 was predominantly present in a long isoform. So it has been suggested that the enzymatic active domain might residue at the terminal axons, and therefore the long isoform be more enzymatically active than the short isoform. And indeed, using a neutral cell background, we found decreased citrullination in the short isoform following exogenous expression of both PADI isoforms, long and short. And if you knock down EWS1, you lose the longer PADI2 version, suggesting that EWS1 not only regulated PADI2 expression, but also PADI2 isoform specific splicing, leading to more short PADI2 transcripts. So functionally, PADI2 knockdown reduces cell cycle regulated genes while inducing a G2M arrest in cell lines. This is data out of RNA-seq experiments after knockdown of PADI2 and the cell cycle experiments after knockdown of both PADI2 and EWS1, showing that knockdown of PADI2 inducing a G2M cell cycle arrest in cell lines. And in a subsequent experiment, we used mass spectrometry to establish the citrullinome, meaning all citrullinated proteins in a couple of Ewing sarcoma cell lines. And if you cross the significant candidates of this assay with the significant altered genes following both PADI2 and EWS1 knockdown, you get two hits, DDX39 and AliRep, which both forms a protein complex called TREX, transcription and export complex. So this complex is involved in transcription, nuclear export, ultimately regulating cell cycle and DNA image. And so overall, we do think that EWS1 uses PADI2 to affect cell cycle proliferation and control RNA biology. And the second compound of our screen, lestaritinib, or SEP701, is a multi-kinase inhibitor with different targets, some of which have also been described in Ewing sarcoma. And lestaritinib has been tested up to phase 2 clinical trials in various diseases, including leukemia, lymphoma, and neuroblastoma. And to the best of our knowledge, data for lestaritinib is lacking in sarcoma, especially in Ewing sarcoma. So therefore, Samit Dayi, a very talented doctoral candidate here in our lab group, treated Ewing sarcoma cell lines with lestaritinib and found reduced colony formation and growth and induction of apoptosis. So he's currently looking into which targets are affected by lestaritinib treatment in the cells. further testing of both drugs using primary cell lines in our CAM model that Wiebke and Max will talk about soon is planned. And we at the Cooperative Ewing Sarcoma Lab hope to provide you with some more data of these inhibitors soon. So with this, I hope I catched up a little bit on time, and I would like to point out that this work could not have been realized without the support of different lab groups around Germany, North America, especially mentioning Thomas Grünewald in Heidelberg, Wolfgang Hartmann Münster, and Jeff Turetsky in Washington. And of course, here in Essen, having a great team led by Uja. So thank you very much for your attention.
Yeah, thank you very much for this great presentation. Any questions? I do not see any questions in the chat here. Is there an urgent question from the audience, which we can? Okay, I do see no additional question. Thank you very much again for this presentation, and we will move on to the next presentation, and we stay with the translation and move to the role of mice in translational research. Didier Surdes will be the presenter. Go ahead.
Yes, thank you. Can you hear me?
Yes, we do.
Yes, so thank you very much for the invitation and the opportunity to present some work. So today I would like actually to share a very long standing question in the field of Ewing sarcoma is what the cell of origin and can we actually modulate this? It has been actually broadly introduced today that this translocation is at the origin of the transformation in the patient. However, actually, we still don't know if this is an event that is possibly occurring in many cells in everybody's, but fortunately enough, we know that fly is toxic. And if this happened, possibly most of the cells would just die from this. So this event is really key to transformation in the patient, but we are really still lacking some faithful model to understand how this happened. And when I mean faithful model, I mean we need to have a primary untransformed cell of origin from a human and recapitulate this event. Now, as mentioned, we know this is toxic, and this has been really a challenge, especially for all the groups that have been trying to do this, for instance, in mice models. And Olivier also really introduced the importance of the genetic background that has been shown through ethnicity studies and more recently through GWAS studies indicating that there are possibly some susceptibility loci, and the genetic background would be very important to generate such model. Now, actually, this project started basically a few years ago due to the synergy that happened at the moment in the laboratory of Olivier, because Thomas Grünewald was doing very early cell culture session to grow up mesenchymal stem cells derived from Ewing sarcoma patient. Olivier had set up a collaboration with Eric Abranet, but was really a pioneer in the world of this molecular scissor. She was working first with Talon and then with a CRISPR tool. And basically, we decided to start a project and try to recapitulate this model using a CRISPR model in a bone marrow derived mesenchymal stem cells using hypoxic condition to facilitate the proliferation of the cells, and use CRISPR guide targeting EWS fly. And using actually small soft agar colonies assay, unfortunately, we were not successful to generate colonies there. Now, beside the genetic background, we know that other events occur also in Ewing sarcoma, and these are just the most frequent event that would be easy to recapitulate using a CRISPR tool, because gain of chromosome might be more tricky, and especially the patient from which this mesenchymal stem cells was derived had a very aggressive phenotype, and he had a deletion of CDKN2A P53, and he had the mutation of STAC2, but he has a very strange skewed expression of the 5' to 3' mRNA transcript, indicating that there might be something wrong at the STAC2 level in this patient. So we wonder actually if we take the same condition and then add some guide RNA together with EW-Fly to recapitulate the translocation, if we'd be able to generate some clones. And actually only when we combine the three guides with STAC2, TP53, CDK, and 2A, we're able to actually generate clones, and we can really see here that there is a benefit of adding the 3 different mutation. So this culture that we turned then EWIMA, and we're able to establish more than 30 different models from this, were grown up to 1 year and really happily grown. And you can also see that in terms of expression of EW-Fly, we have similar expression level to the typical A673 cell line, although we also had some models with a lower expression of EW-Fly. And you can see in this model, STAC2 expression is lost with a truncated P53 and also a loss of P16 expression. Now, these events really activate the telomerase activity, as you can see here using a trap assay. You see the parental cells, the MSCs from this patient, and you can really see through this assay that the more pattern, the ladder you see, the more telomerase activity is present in these cells. And you can see here A673 and all these EWIMA clones, and kind of reduced activity for the clones where you had the reduced expression of EW-Fly. Now, a key feature, and if you want to generate a faithful model of Ewing sarcoma, is of course its pioneer function and its ability to bind to ETs or these microsatellites. And for this, basically, we perform ChIP-seq with these cells. And you can see here the prototype locus, the NKX22. This is completely closed chromatin in these mesenchymal stem cells. And you can see binding of EW-Fly and these neon-answer formation, which are very similar to the one that, for instance, of A673 cell line. We also know that EW-Fly can repress, although the mechanism is not fully clear how this functions, but clearly here you can see at DKK1, sorry, that upon binding of EW-Fly, this region tend to compact here. So this is just a summary of the binding property of EW-Fly genome-wide, and you can clearly see that the EWIMA cells here resemble A673, and the binding pattern of EW-Fly is very different from the parental mesenchymal stem cells. And to do actually motif calling, the first one that is identifies is the expecting GGA microsatellite, and the second one is an ETS binding site. So really, this is the key and classical feature that you expect from a Ewing sarcoma model. Similar at H3K27 acetylation level, you also clearly see that the pattern of EWIMA and A670 resemble, and chromatin is closed at the region that were activated mesenchymal stem cells. And again, calling some super enhancer there, you can find in this model the expected candidate that had been actually previously published. Now, using this cocktail of guides, basically what you do, you cut the genome of your mesenchymal stem cells, and you can generate translocation as shown here through a karyotype profiling, and you can also induce point mutation in CDKN2A, TP53, and STAC2. So that was the case for part of our model, but more surprisingly, we found some model actually with this kind of ping pong pattern, where basically the EW-Fly one connection was fusing with another region than TP53, CDKN, and 2A. And actually, this is a very reminiscence of a mechanism that is called, chromopexy, which basically was described a few years ago from Anderson, the group of Adam Schleen, and is a balanced translocation that basically reshuffles multiple chromosomes into new scramble configuration, and usually this involves more than 3 chromosome. It drives, it doesn't alter the copy number in these events. And actually what has been described is that these loops preferentially occur in early replicating and transcriptionally active genomic hubs. And actually, even though our system is of course artificial to generate this kind of chromoplectic pattern, it was quite striking that we were able to generate this chromoplectic pattern from the guides that were cutting in early replicating region, including this region here, which was an off target of the CDKN2A guide, whereas actually the guide that was hitting STAC2 only generated a point mutation in this gene. Now, the key experiment, of course, we have generated cell line that propagates nicely in vitro, and that are immortalized is, do these cells actually transform and can generate tumor in mice? And here we injected orthotopic cells into the femur of NSG mice, and we also check if these cells were able to generate some distant growth of the tumor. And to make the long story short, actually we tested 3 models, and within 2 months we could nicely observe tumor grow in these cells. You see the classical small round cell pattern with STAC2 loss, highly proliferative membrane CD9 expression, and we could also detect some distant growth from these primary injected cells in the leg of the mice, in the lung, and also in the liver. And again, in vivo, these cells were expressing similar EWS-fly level as the parental cells in vitro. We finally characterize these models using RNA-seq, and here is a PCA representation, and I hope you can appreciate that basically the first component has a really majority weight as compared to the other component, which is presented here. And basically these principal components is where you see the mesenchymal stem cells. If you add only the STAC2, P53, and CDKN2 mutation, this does not alter the transcriptional profile. But whenever you add this EWS-fly SPC, you have a major shift towards where you see Ewing sarcoma cells, and putting these cells in vivo shift them towards even more Ewing nest state. Whereas if you silence EWS-fly in this model, you go back to the right. And the model that I mentioned that had a lower EWS-fly activity were really also lower in this representation. So just as a signature, we also showed that what you expect is what is enriched is an EWS-fly activation signature that is really present in this model, as well as other known EWS-fly associated signature. And on the other side, what is expressed in a mesenchymal counterpart is the typical ETS downregulated signature, as well as this EMT transition that now we know it's not an epithelial, but it's a Ewing to mesenchymal transition, but the player may be quite similar. And quite intriguingly, so morphologically, you can see that you have MSCs with this mesenchymal pattern, giving rise to these Ewing cells models and tumors, but you also had these intermediate models which had again an intermediate morphology. So what I just show you today that we're able to generate de novo Ewing Sarcoma, but we think is a faithful model starting from Ewing Sarcoma, derive mesenchymal stem cells and generate mouse tumor. What I didn't show you for the sake of time is that we did this also with ES mesenchymal precursors derived cells, but actually we never managed to transform them and generate a stable cell line with this, indicating that possibly that the cell of origin indeed and the genetic background is important, but would be needed to be recapitulated in many more models really to make sure that this holds and is true. Now I would just really thank here. This was a fantastic collaboration with the team of Erika Brenet, and most of the experiments have been done by Anna Sole, postdoc in the lab. And of course, this wouldn't have been possible with the incredible infrastructure that we benefit in the lab of Olivier. And I would also thank really Sandrine Grosstedt, Sakina Zaidi, Cécile Perugène, and Isabelle here. And also thank again Thomas for initiating this project around mesenchymal stem cells, and of course patient and family, and all charity that funded this work. Thank you very much.
Yeah, thank you very much, Didier, for this interesting presentation. There is a question here appearing. Is the level of fusion important in determining the transformation capacity? The level of fusion important for transformation capacity? We have a few models. We have these 4 models that have a lower expression of the fusion oncogene, but we didn't have a chance to inject them into the mice. They were growing quite happily for like 2 to 3 months, but would need still some work to just grow them for a longer time, and also try to inject them into mice. But yeah, this is a very interesting question, and unfortunately the COVID made this kind of experiment a bit more tricky, but definitely something that should be tried. Yes. Yeah, thank you very much again. So we stay with animals from mice to, so I do see a cat on the screen here, and we will move to eggs, and there will be the next presentation on chem assays given by Wiebke Guder and Maximilian Kerkhoff.
Yeah, thank you very much. Today it will just be me presenting. I hope that's fine either. Okay. Hi, everyone. I'm happy to be able to have a little talk about my project today, which is a cooperation between the groups of Uta Dirksen and Wiebke Guder. And we are currently working on a CAM model for evaluating therapy responsiveness of cell and patient-derived sarcoma tumors. I will quickly turn on the laser pointer. Okay. The CAM, short for chorioallantoic membrane of fertilized chicken eggs, is an excellent matrix for tumor engraftments because of its strong vascularization. Of course, every model has its limitations, but the CAM model is an intermediate between cell culture and mouse models. On the one hand, we have low costs, rather low amount of effort, and a high throughput of samples. On the other hand, it has a very high engraftment success rate and a tumor environment which can remodel the situation within the patient to a certain extent. However, its biggest disadvantage may be the rather short time of 1 week the tumors can be observed because of the fast development time of the chicken embryo. So to test the model for sarcoma, we cut a window into the eggshell on developmental day 3 and sealed it again with a tape. And at day 10, we opened up again the egg and perforated a blood vessel with the scalpel and applied either tumor cells from culture or fresh tumor tissue obtained from patients. And initially, we engrafted several different sarcoma entities also from different origins, to evaluate engraftment capabilities of the CAM. And interestingly, we were able to grow cell-derived tumors from all tested entities. And these different entities presented different morphologies. Some were growing a little more flat and some a little denser. And also there were different tendencies to hemorrhage between the tumor entities. And furthermore, we were able to successfully engraft most of the tumor patient tumor tissues, and even tumors that were hard to culture grew on the CAM. So this really convinced us of the model. Now we wanted to go a step further and ask ourselves if we could employ the model to evaluate whether we can make use of photodynamic diagnostics in sarcoma treatment. 5-aminolevulinic acid is the natural substrate in heme biosynthesis. And in cancer cells, protoporphyrin 9, which is the pre-step of heme, is processed into heme very inefficiently due to the Warburg effect and the resulting low pH. And therefore, there is no negative feedback loop in the endogenous 5-aminolevulinic acid synthesis. And upon administration of additional 5-ALA, protoporphyrin 9 accumulates within tumor cells. And this accumulation can then be exploited as protoporphyrin 9 will emit red light when excited by blue light, which can be used for photodynamic diagnostics. And this mechanism is already used, for example, for fluorescence-guided surgery of bladder cancer and glioblastoma. And to test this approach in Ewing sarcoma, we treated both tissue engraftments and cell-derived tumors with 5-ALA for 4 hours and read out the fluorescence after blue light excitation with an endoscope. And for most of the tumors, we saw a specific red fluorescence of good intensity, suggesting a potential use in fluorescence-guided surgery also for sarcomas. Another big part of our research is to establish a model for the evaluation of therapy responsiveness. And our concept includes 3 main branches. First, we want to generate cell models from patient tumor material. And second, we want to apply these cells and also, if possible, directly tumor material onto the CAM. And in both cases, we want to perform drug testing with both established as well as new substances and characterize the changes on RNA, protein, and histology level afterwards, hoping to identify subgroups of good, bad, and non-responding tumors. And furthermore, we collect blood samples from patients to investigate possible markers in the blood that might correlate with the tumor aggressiveness and response or non-response to chemotherapeutic agents or other substances. By this, we are aiming to enable a more individualized therapy approach. So basically, we are adding 2 steps to the protocol. First, we treat the tumors with a substance, and second, we read out the changes after treatment at different levels. And initially, we want to establish a fast and simple readout for tumor vitality as the first indicator for treatment response, to make sure of consistency in the results. And the easiest way to track this is the size reduction of the tumor. And it was already shown before that, for example, in ovarian carcinoma, the tumor size on CAM could be reduced 3 days after doxorubicin treatment. However, for Ewing sarcoma, this seems not to be the case, as we were not able to reduce the tumor size even with very high concentrations of doxorubicin or vincristine. Therefore, we came up with the idea to use the acridine orange ethidium bromide co-staining, which is well established to detect and quantify cell death in cell culture. And here you can see the color shift from vital cells, which appear green, to vincristine treated dying cells appearing red to orange. And in the first attempt, we stained tissue engraftments that did not work too well, and saw red fluorescing loose tissue and a more greenish tissue, which attached a little bit better to the CAM. And preliminary results for untreated and treated cell-derived tumors, we saw a nice color shift from green indicating a vital tumor to red indicating a dying tumor after vincristine treatment. So the next steps will be to validate these results and to determine concentrations for chemotherapeutics and other substances for use in our model. And with this, I want to end my talk today. And yeah, thanks for your attention.
Thank you very much for this very interesting presentation. I had a question. As a pathologist, how long can you keep those tumors on this CAM membrane? And do you have an idea how the tumor changes simply by placing it on this CAM membrane?
Yeah, unfortunately, we have just about 1 week time, because afterwards it would count as an animal experiment, and that's one of the big advantages, that it's not counting as an animal experiment until day 18. So until day 18, we have to terminate the embryos. Therefore, we have just 1 week for the tumor to develop. Yes. And sorry, what was the second part?
The second question was, how does the tumor change simply by placing or by being kept on this membrane? I think of the
you mean the cell-derived tumor or the patient-derived tumor?
The patient-derived tumor.
Okay. I think Wiebke can explain this a little bit better, because I don't have so much data seen so far yet from patient-derived tumors.
Would you like me to step in?
Yeah, please.
So what we have done to begin with was that we used patient-derived tumors, and the cycle of the egg development takes 21 days, and you open the egg on day 1, you apply the tumor on day 10, and you harvest what you sowed on day 17 to 18. And so the period is approximately 7 days that the tumor stays on the CAM. If you have patient-derived tumors, what you do see is that not all of the tumor will attach to the CAM and will be vascularized. It is mostly the lower levels more closely located to the CAM that will be vascularized and that will thrive on the CAM. The areas that are more distant to the CAM have the problem that they are probably not receiving enough nutrients to survive, so they form a high rate of necrosis. And this rate of necrosis we can see pathologically. So we also started using cell cultures, using Matrigel to be able to stop the tumor from spreading on the CAM and to form a tumor. And in these cases, you see that the Matrigel is, taken over by the tumor cells, but they build little bubbles in the Matrigel, and the longer they stay on the CAM, the more Matrigel is changed into the tumor cells.
Yeah, thank you. Very interesting. Thank you very much. So I do not see any additional questions. So that means we will move on to the next presentation, which will be presented by Heinrich Kova, and is entitled "Networks of EWS Fly."
Okay. can you see my presentation in the presentation mode?
Yes, we do.
Okay, great. So Uta asked me to talk about networks. And since this is a symposium organized to memorize the 100th anniversary of the first description of the diffuse endothelioma of bone by James Ewing, I thought I start maybe with a little anecdote from James Ewing's life. And going back another 30 years in history. So here in this picture, you can see the girl on the right-hand side is Bessie Dashiell. She was a 17-year-old girl who after an accident was diagnosed with a small round cell sarcoma at her humerus. She was a very good friend of John D. Rockefeller Jr., so the son of one of the most influential and the richest families in the US at the time. And maybe that's the reason why she got operated with her tumor by the chief surgeon of the New York Cancer Hospital, William Coley, who amputated her arm, but yet could not save the girl. And he felt so sad about the death of that beautiful young girl, young woman, that he decided to dedicate his whole professional and scientific life to the treatment of sarcoma patients. For this purpose, actually, he invented a new treatment modality. He actually started exactly 130 years ago for the first patient to inject Streptococcus pyogenes into the tumors, so bacteria into the tumor of sarcoma patients. And he published several papers reporting successes with this kind of treatment, initiating inflammation. And that's why he's considered to be the father of immunotherapy. And he got very famous for this kind of treatment, which was known as Coley's reagent. And for that reason, he was actually a candidate for the directorship of the New York Cancer Hospital. But for that position, he was competing with James Ewing. And James Ewing also had his network. And in his network, there was James Douglas, the CEO of Phelps Dodge Mining and Coal Producing Company, a big company, big mining company in the US, whose daughter suffered from a breast cancer. And as we have heard already from Paul Mayers this morning, James Ewing was a big believer in radiation therapy, which at that time was not yet available at the New York Cancer Hospital. And for that reason, he not only proposed, but actually accompanied in person the young woman to London to receive radiation therapy. And even though, again, the patient could not be saved, James Douglas, out of gratefulness, promised to donate a large amount of radium, actually half of what was available in the world at that time, to the New York Cancer Hospital under one condition, and that was that James Ewing should get the job as the director of the New York Cancer Hospital. And that's what happened. And when Ewing became the boss of William Coley, he stopped him from treating patients with this Coley's reagent because he had serious concerns due to reports on problems about the studies not being well controlled. So this set an end to immunotherapy for decades to come. All right, so now let's talk about Ewing sarcoma. I think we all agree that pediatric cancer is caused by some rare mutations that block the normal development of an undifferentiated, most likely embryonal cell type of origin on its way to a differentiated tissue. And it is the specific combination of the mutation and the molecular environment and the tissue environment at the time of the perturbation of the normal development that is responsible for the expression of the full set of cancer hallmarks and the specific characteristics of a cancer from age of onset to therapy response. And so it is clear that if we understand the normal development as well as the perturbed development of that enigmatic cell type of origin, we should be able to model the disease and accelerate treatment innovation. Now we know a lot about the mutation already, and EWS-FLI1 has been described already by several speakers during this symposium. So just to add here an aspect, and that is that EWS-FLI1 is actually perturbing gene regulation, genome-wide gene regulation by several different mechanisms, depending on the partners and the specific chromatin context. So EWS-1 can bind to consensus its binding sites as a monomer, either alone or together with other transcription factors in an activating or repressive mode. It can also act as a transcriptional co-activator with glucocorticoid receptor, for example. And as we have heard, it can phase condensate and form multimeric complexes binding and opening up GGA microsatellites, an overactivity of an oncogene that has not been described for any other oncogene in any cancer so far. And there is data from several labs, including our own, that show that actually different levels of EWS-FLI1 have different phenotypic consequences. If EWS-FLI1 is high, the tumor cells are proliferating. If it's decreased, then they adopt a more migratory and invasive phenotype. And if it's too low, then the cells may die. And this may be due to the different mechanisms by which EWS-FLI1 deregulates the transcriptome require or occur with different affinities to the chromatin, and therefore depend on different thresholds of the protein. To study this in more detail, we have recently, and this is the work of Valérie Fock and Vivian Suresh in the lab, we have inserted a fluorescent tag and a degradation tag into the 3' end of the endogenous EWS-FLI1 gene in 3 Ewing Sarcoma cell lines. The data that are shown here are from 1 cell line, A673, 3 different cell clones. And now if we add different amounts of the DTEC ligand, a small compound that leads to the degradation of the protein very rapidly, within 1 hour, we can degrade the protein much faster than with an H or an siRNA. And we can establish different thresholds of the protein. You can see that even a slight decrease on this side here, a slight decrease of the EWS-FLI1 protein has already profound consequences for the expression of target genes, both on the repressed target genes as well as activated target genes. And you can see that even specifically for those genes that are regulated by GGA microsatellites, the response is very strong, even with a slight EWS-FLI1 fluctuation. So far about the fusion protein, but what about the tissue context? And this is something that over time, since the first description by James Ewing, many different approaches have been taken to identify the cell type of origin and the developmental context. Histology, immunohistology, and functional genomics by knockdown of EWS-FLI1 mostly indicates, as we have heard, that it's most likely deriving from some mesenchymal cell, mesenchymal stem cell, either of mesodermal or neuroectodermal origin. There is single cell omics, which is still in its infancy, but most likely able to identify the specific stage of differentiation much more precisely at which EWS-FLI1 is active. And then there is animal modeling. And many labs have embarked on animal modeling years ago already, and everyone failed so far, including our own. But still, I'm going to tell you a little bit of our own attempts, even though this has been a dead-end road so far, and no animal model, genetic animal model for Ewing sarcoma has been developed so far. Okay, so Barbara Sachs, about 15 years ago in Richard Morley's lab, a PhD student in Richard Morley's lab, developed an EWS-FLI1 knock-in mouse model. EWS-FLI1 was knocked into the ROSA26 locus, and it was activated with a mesenchymal CRE, the PRX1 driven CRE that targets EWS-FLI1 to the mesenchymal lineage of endochondral bone formation. So you can see the expression of EWS-FLI1 in the limb here, for example. This resulted in profound skeletal malformations, mostly due to the fact that the bones could not ossify. They are arrested in an early chondrocytic stage. The mice die around birth, they don't develop tumors. However, one can isolate those cells from those fetal limbs and propagate them. They are immortalized by EWS-FLA1, but still not fully transformed. If you put them into soft agar or into mice, they will not give colonies or tumors. They express EWS-FLA1, as you can see here. So this is the work of Rahil Noor-Sadegh, a PhD student in the lab that I'm going to describe to you in a bit more detail in the next few slides. So she wanted to know what's going on with these cells. Why are they not transformed? What is missing? And so first, she performed RNA-seq and ATAC-seq, so looking for differentially expressed genes and differentially accessible chromatin regions. And to our great satisfaction, it turns out, and this is done by Florian Halbritter, a computational biologist in the institute, we see an enrichment in GGE microsatellites, which is what we would expect if EWS-FLA1 is functional. And also very satisfactorily, we see an enrichment of a typical Ewing sarcoma EWS-FLA1 signature. So what's going on here? So we were thinking about when actually does Ewing sarcoma occur? Because so far we are looking to fetal cells, but Ewing sarcoma, as we all know, occurs primarily during adolescence, during puberty, at the time when there is the growth spurt, when bones are growing. And in fact, during this period of time, there are several hormones increased. And specifically, I would like to draw your attention again to IGF-1 that has already been addressed in several talks today, because IGF-1 is locally produced mostly by osteoblasts and osteocytes in response to mechanical load in the growing bones. Its mechanical stress is stimulating IGF-1 receptor signaling via focal adhesion kinase and also other signaling pathways like RhoA and also YAPTAZ TEAD signaling. There's also some sparse evidence for an epigenetic activity of IGF-1 from multiple myeloma, for example. So the question was, could IGF-1 that comes from the microenvironment in the growing bone play a role in full transformation of EWS-FLA1 immortalized mesenchymal stem cells, bone marrow mesenchymal stem cells? The role of IGF-1 as an autocrine growth factor is very well established in Ewing sarcoma. Starting, in this publication has already been mentioned, but mainly based on the pioneering work of Jeff Turetsky and Katja Skotlandi even from the 90s already, that showed that if you block IGF-1, or if IGF-1 receptor is not present, if you block IGF-1 receptor, if it's not present, then you interfere with the sustained growth of Ewing sarcoma cells. And that is mostly due to the fact that EWS-FLA1 is generating an IGF-1 signaling conducive cellular environment. It activates the IGF-1 promoter, it suppresses IGF-1 receptor signaling modulatory microRNAs, and it upregulates papalysin, which is a protein, which is an enzyme that degrades the negative regulator of IGF-1 signaling IGF binding proteins in the immediate microenvironment of the tumor cells. However, as we've heard today, despite there were some promising results from in vitro and xenograft experiments using IGF-1 receptor blocking antibodies, we have heard already, and this is data from a meta-analysis of several studies that have been clinical trials that have been performed, only about 10 to 40% of Ewing sarcoma patients respond to IGF-1, anti-IGF-1 therapy. So we wanted to investigate the role of IGF-1 in tumor initiation and not in sustained proliferation of the tumors. And so Rahil took this EWS-FLA1 expressing mesenchymal stem cell-like cells, as we call them, which are immortal, as I told you, into soft agar. And as I told you, no colonies are forming because they are not transformed. And now she treated these cells for 4 to 5 weeks with either IGF-1 or insulin or a combination of both. And now she starts to see a lot of colonies growing in soft agar. And that was very significant. Excitingly and intriguingly, when she now took those cells from the soft agar colonies and expanded them further in the absence of any hormone treatment and assayed them again in soft agar assays, without any hormone. So these cells are already 5 weeks without any hormone treatment. They retained their transformed phenotype. They formed colonies very nicely in soft agar, and you can take those cells actually and inject them into mice, and they will form tumors very efficiently. So that means that the transient treatment with IGF-1 or insulin or a combination of both stably reprogrammed those cells to a fully transformed phenotype. And this was still dependent on the expression of EWS1, since knockdown of EWS1 there's a greatly decreased colony number. I see here a yellow line that don't know where it comes from. I don't hope that you see it as well. Anyway, doesn't mean anything. Okay, so she now again asked the question, what is the difference between the EWS1 expressing immortalized cells, parental cells, and the IGF-1 reprogrammed cells? Again, performing RNA-seq and ATAC-seq. So from the RNA-seq, the first thing that she observed was that there is an enhancement of the EWS1 signature, both for activated genes as well as for repressed genes. However, there was no increase in the enrichment of GG microsatellites, but rather there was a little decrease in the enrichment of GG microsatellites in the open regions in the hormone reprogrammed cells as compared to the EWS1 expressing parental cells. So Florian Halbritter, the computational biologist, performed a very detailed analysis of the ATAC and RNA-seq data. This is a very complex slide, but just bear with me, I'm just guiding you through it. Actually, we just have to focus on the left part here of that figure, where he performed clustering of the differentially accessible chromatin regions. And he could cluster them into 5 different modules, as you can see here. And I would just like to draw your attention first to this cluster, this small cluster M2, which comprises chromatin regions and genes which are closed in the wild type MSCs, independently of whether they were treated with IGF-1 or not, and also closed in the EWS1 expressing mesenchymal stem cell-like cells from the knock-in mouse, from the EWS1 knock-in mouse, but opened up upon reprogramming with IGF-1. And this group of genes comprises a number of very interesting genes that we know very well from human Ewing sarcoma. Bbcl that Thomas Grünewald has described as a hallmark of Ewing sarcoma and being even more specific than CD99 and other markers of Ewing sarcoma. Protein kinase gamma B that Olivier has been talking already about. R-spondin-1 Wnt signaling receptor. Very importantly, SOX2 that has also been addressed already during the course of this symposium today. And very interestingly, YAP, which is a transcriptional co-activator of TAD transcription factors. And laminin 5 and RH42 are genes that are somehow related to YAP, and don't want to go into details of that interesting connection. But just keep in mind that we see YAP coming up here. And that is interesting because the module 4 that is shown here, that comprises chromatin regions that are opened up only in the EWS1 expressing situation independently of IGF-1 treatment, comprises a set of genes which are yet differentially expressed between those 2 different conditions. So here are the cells that express the parental cells of the knock-in mouse that express EWS1, and here are the cells derived from them after IGF-1 reprogramming. And still there are genes that are expressed here, but not here, or much lower expressed here. And this group of differentially expressed genes includes also cyclin D1 and KLF5. So this was very interesting because it means that it needed IGF-1 to activate those genes, although the transcription factor TEAD, either 1, 2, 3, or 4, and EWS1 might have been already sitting there and opening up the chromatin together with EWS1. I see the number of yellow lines here increasing. I don't know what's going on here at all. I hope you don't see that. I don't know what it means.
This is related to the time, I guess.
I see. Okay. All right. Okay. So I just wanted to say that we have seen previously that actually EWS1 binds a large proportion of TEAD-bound genes in human Ewing sarcoma. Actually, more than 80% of TEAD-bound genes are bound by EWS1. And we've published this already previously. And so the model that we are currently pursuing and try to functionally validate is as follows. We think that EWS1 is opening up regions that bind to TAD together with EWS1, which is not sufficient to drive gene expression because it still needs the binding of the transcriptional co-activator, YAP, which is only activated upon IGF-1 reprogramming through a mechanism that is still not clear. We have some ideas about that, but it's much too early to talk about that. And other genes that are related to YAP, and also, and that will activate a number of important genes like cyclin D1 and KLF5, which are induced, which are involved in the self-renewal of tumor cells, together with SOX2 and together with BCL11B and others that are activated in an IGF-1 dependent way by EWS1. So that's our current working model. And with this, I'm going to, yes, just to say, if this would be the case, then blocking the association of YAP with TEAD using an inhibitor, a small molecule that is actually binding to the palmitoylation pocket of TEAD, should actually decrease colony number in the IGF-1 reprogrammed cells. And that was actually the case. As you can see here, we see a concentration-dependent decrease in colony formation, soft agar colony formation upon treatment with this YAP-TEAD inhibitor. All right, this story needs to be continued, and I hope I can do this the next time when we meet in person, hopefully. And I would like to thank the people involved in the wonderful team that I currently have in the lab that are shown here. And specifically, I would like to thank Rahil, who did most of the work that I've presented to you, Valerie and Vivian, and Florian, the computational biologist, and many other people involved in the projects in the lab, and the many people, and this is just a start of the list is much longer, of people who have been accompanying us in our work for many years, and the funding organizations. And thank you for your attention.
Thank you very much for this comprehensive and excellent presentation. Thank you very much. There is one question in the chat, so I would like to ask you to give a very short answer to the time. The question was on, did you test the contribution of these events such as STAC2, TP53, CDKN, and 2A mutations, for instance, together with IGF in your model?
No, we did not. We did not introduce any other mutations. We just introduced EWS1.
Okay. So thank you very much again for this excellent presentation. And we will continue to the last speaker in this session, who will be Enrique de Alaba Casado. And he poses a question: what do pathologists think about CARs? So I'm very curious to learn the answer.
Yeah, okay, thank you very much. Can you hear me and see my presentation?
Yes, excellent.
Okay, so first of all, I would like to come back to history and to claim a little bit the legacy of James Jewin as a pathologist. He was not only a prominent pathologist in his time, as we have learned from Paul Meyers and Heinrich Kovar, but also he was a great and novel in trying to describe a new entity, not only by its morphology, but also by its exquisite sensitivity to radiotherapy. This combination was a novel concept at that time, and means that pathologists at that time were really concerned about innovative therapies. And so we are now today. So that may be the reason why Uta asked me to try to answer to this question: what do pathologists think about CARs, okay? So CARs, immunotherapy, maybe we are coming back to Foley's liquid, are very famous already in hematologic neoplasms. But what about solid neoplasms? So you know that when you see objects in the mirror, maybe they are closer than they appear. And they may be meaning that we are actually getting to the time in which we have to think about CARs also in solid tumors, especially in Ewing sarcoma. So let's think about, for the sake of people from the patient organizations, I found a very simple cartoon on CAR-Ts. You know, CAR-Ts are T cells that we isolate from one patient, and we engineer to express a chimeric antigen receptor, so a receptor that can, to engineer a T cell that can recognize a specific receptor or a specific antigen in one particular tumor cell. We can expand this population and infuse that into the patient. This is the promise, and this is good. But then thinking about Ewing sarcoma and trying to answer what do we pathologists think about this, I think we have to face two problems. First problem has actually been raised in one of the talks before is, are there specific cell membrane antigens in Ewing sarcoma? A specific meaning not present in normal tissues. And if you see, you think about extracellular targets, there are many famous molecules that also are present in normal tissues. So think about CD99, for example, which would be an excellent candidate. It's present in Langerhans cells, T cells, and many other normal cells. And the same for other fantastic candidates. So we and others have been doing single-cell RNA-seq of several genes and antigens in human healthy tissues and healthy bone marrow and peripheral blood and MSCs, and found that many of the famous candidates are not safe. Are not safe enough. We have been talking about CD99, IGF-1R, and other candidates. They are basically not safe. Even in the case we were able to find a safe target, imagine Olivier Delattre finds his neo-genes and neo-antigens and in the memory of the cell, then the question would remain, are they effective enough? And then to try to solve this, we pathologists have to do something like trying to see the expression of this in the tissues. And this is a lot of work. When I think on this, I think on constructing a big tissue microarray with Ewing sarcoma samples to try to demonstrate expression in a big array or in a big series of cases. And not only that, also do the immunos and try to take the evaluation, get spreadsheets with data, and try to get the expression of antigen A or antigen B, whatever it means. This is a huge problem. This is something we think when we hear about CARs in Ewing sarcoma. This is problem number one. And problem number two, as pathologists, we think that Ewing sarcoma stroma is a cold desert. We know that tumors have not only tumor cells, but also stroma. We heard about microenvironment today. But this is an immunological desert because they escape immunity. The low mutational rate in many Ewing sarcomas, not all of them, but many of them. HLA class 1 is very heterogeneous, no class 2 molecule expression. We have heard about PDL1, there's no consistent PDL1 expression in Ewing sarcomas. And the cytokine and chemokine profiles in the microenvironment is not clear. So we are basically facing a situation in which T cell response, antigen presenting cells could activate T cells and could target the cells. It is not the case in Ewing sarcoma, in which we have lack of, again, HLA2, class 2 molecules, and lots of myeloid-derived stem cells, fibroblasts inhibiting Tregs, inhibiting the response of T cells. So how can we get rid of that? Later today, we will hear about the next things ahead. So in the next blog, we will hear about the work of Claudia Rosic on GD2 CAR T cells, and on Stefan's and Bbcl talking about something close to that, engineered TCR T cells. So I'm eager to listen to that as a pathologist. But what can I think on that? Or how should I think I could overcome this? So we've been thinking on that, not only in the lab, and in collaborations nearby, and with several of the labs present in this conference today. And for example, for the first thing, okay, in terms of increasing safety, this is the usual structure of a CAR T, so having the effector domain close to the co-stimulatory domain, going to the same antigen recognition sequence. Why don't we prepare, for example, dual CARs in which we have the effector domain close or linked to one of the antigens and the co-stimulatory domain to the other antigen, meaning that unless you have both antigens expressed in the tumor cell, you will not get an active response. This could increase a lot, I think, safety, unless Olivier comes with a neoantigen in the membrane. And maybe, I don't know whether this could hinder effectiveness of this design, but I think this could be a possibility to test and to try. And the other thing, thinking on the second problem we find, I think we should prime the microenvironment, meaning that we have to heat it up before the CARs enter the tissue. One possibility, of course, is trabectedin, one of the drugs inhibiting some of the inhibiting molecules in the microenvironment, and I think we'll hear about it in the next block of talks again. And of course, we can think of VEGF inhibitors or overexpression of some pro-inflammatory cytokines. This is just two simple proposals to think about. And maybe in the next year, we will have a all new 2022 CAR. So I was intended to be very brief, coming back to immunotherapy, and thanks to not only my group with a yellow label of Sarcoma Awareness Month, but also with collaboration with great people like Pablo Menendez, coming from the world of CARs in leukemias, and also Claudia and Katia and Thomas and Angel, with we have been thinking and maybe preparing a nice proposal for the future. Thank you very much.
Yeah, thank you very much for this interesting presentation. So pathologists may consider different things when talking about CARs. Yes, of course. Do you have so honestly, one question. So you described this specific stroma in Ewing sarcoma as a cold desert. So I think this is what we are seeing there. Do you see any? I think there are also ways to influence the stroma or to target the stroma, to change the composition of the stroma. Do you any approaches to this in terms of Ewing sarcoma as well?
Yeah, that's what I tried to mention before. So I think we have to prime the stroma. We have to heat it up. We have to make it inflamed or hot. And maybe, again, some drugs can be helpful to prime the stroma. Specifically trabectedin, we have some experience together with Katia and other people in the audience, Pat as well, of course. So I think this could be a good combination, and there are, of course, other combinations that could be considered. So, but I think we need to study in depth the stroma of Ewing sarcomas.
Do you see the changes in the stroma, or how the stroma changes morphologically?
No, not yet. This, we have done some in vivo work and some in vitro work, but of course we didn't do any work in humans. But some of the models suggest that this could be working.
Okay, Very good. Thank you very much.
Thank you.
So with this, I do not see any additional questions in the chat. So with this, I would like to close this session here. I would like to thank all the speakers again, presenters here. So we had a very good, very interesting session here. And with this, I hand over to the next chair, who will be Sebastian Bauer for the next session. Thank you very much.